Action potential arrives at synaptic knob. Calcium channels in membrane open, allowing calcium to move into the cell. Increased calcium causes synaptic vesicles filled with acetylcholine to fuse with the membrane. Acetylcholine is released from the synaptic knob by exocytosis into the synaptic cleft. Acetylcholine is picked up by receptors on the motor end plate of the skeletal muscle. Sodium-potassium channels open, allowing sodium ions to enter the muscle fiber. Repolarization occurs, and potassium goes into the synaptic cleft. An increase in positive ions inside the muscle cell initiates an action potential to travel on sacrolemma.
After cycle is complete, acetylcholinease repolarizes, bringing muscle back into relaxing state.
Action potential travels down sacrolemma into T tubules and stimulates the release of calcium. The calcium comes from the terminal cisternae of the sacroplasmic reticulum, where it has been sequestered by calsequestrin, into the sacroplasm of the muscle cell. Due to calcium overload in the terminal cisternae, calcium travels down and floods over the thin filaments and binds to troponin, pulling tropomyosin off the active site on the actin. High-energy myosin heads leap to bind on the exposed active sites on the thin filaments, forming a cross bridge. The myosin head pivots to low energy state, releasing ADP, and moving the thin filaments over the thick filaments, the power strip. ATP binds the myosin head, causing the head to break free from the active site on the actin. The myosin head becomes energized from the mitochondria as ATP is converted to ADP. Calcium is pumped back into the sacroplasmic reticulum and attached to calsequestrin, allowing tropomyosin to once again cover the active site, resulting in relaxation of the muscle. A-bands are where thin and thick filaments intersect, and the I-bands are only where thin filaments are found. The Z-disc is where one sacrolemma ends and another begins, leaving the H-band the only place to find thick filaments. This is an example of the muscle fibers being contracted and relaxed. This is an example of a single myofiber. An extended fascicle shows the multiple myofibril within.